Hi everyone, and welcome back to my channel. So in this video, we are going to go through sort of an introduction to Mendelian genetics, remind ourselves about the key concepts. I do have at least one other video on Mendelian genetics if you want to watch that one as well. That one is not voice over PowerPoint. This one is. So let's get started. So when we talk about Mendelian inheritance, we're talking about the result of the work that Gregor Mendel did back in the mid 19th century, using his pea plants and using those as a way of understanding how the process of inheritance worked. In one of my other videos, I do talk about sort of the whole background of that story, how he did his experiments, and that sort of stuff. So I would encourage you to take a look at that one as well. In this one, I'm going to kind of just go through the results and then what we know about how it relates to our modern understanding of genetics, because while Mendel discovered several underlying truths about inheritance, there were some things that he just got wrong. So let's start by going through the 4 laws of Mendelian inheritance. And I'll just pause to remind everyone that Mendel was a monk, not a scientist. And also in biology, we're not allowed to write laws for things because that gives us sort of a sense of undue certainty about the biological world. And as you're going to see, pretty much all of these laws end up being broken by something that we now know happens during with respect to inheritance that Mendel never discovered. So be careful with the use of the words, the use of the term laws when we're talking about biology. But his first one was pairs of genes. So in his understanding, each characteristic, so something that you would describe about an organism, is controlled by 2 discrete inherited units. Okay, that was his terminology. We now know those inherited units to be called genes, and these genes are passed from the parental generation to the offspring. This was based on his understanding that essentially each characteristic has a pair of genes, and that's how it works. We now know that that's not always the case. The second rule is his law of dominance. Traits can either be recessive or dominant. In the case of a heterozygous organism, which is an organism that possesses one trait of one type, one trait of another, the phenotype or the outward appearance of that trait is controlled strictly by the dominant trait. That would argue that if you have both, one's dominant, one's recessive, the dominant one always controls the phenotype. We also have Mendel's law of segregation. What this basically means is, given that the offspring have an equal chance of inheriting, so both parents, mom and dad, both have 2 copies of each gene that controls a characteristic. Okay, that's pretty simple, right? Well, what the law of segregation tells us is that you have a 50-50 chance of inheriting either of dad's alleles for a gene, versions of a gene, or a 50-50 chance of inheriting mom's. So basically, it doesn't, like, essentially there's no like favorability that dad's going to give you his paternal chromosome moreover than his maternal chromosome, and mom's going to give you her maternal chromosome versus her paternal chromosome for a given trait. Basically, this still holds up. Essentially, what this means is, and what Mendel boils this down to is, genetics is all about probability, and he's not entirely wrong on that, and we'll learn more about that later. And then his 4th law is the law of independent assortment, which basically says the inheritance of one gene is not dependent on the inheritance of another. They're all independent. So your gene for eye color would be inherited completely separate of what happens with your hair color, which would be completely separate of what might control whether you have a widow's peak or attached earlobes. And we'll actually find out that this isn't strictly true as well. So all told, 3 out of 4 of Mendel's laws of inheritance end up being broken in the end. And we'll talk about this as well in this video, as well as in subsequent videos. So let's talk about the pairs of genes. Let's break down the first law. Each characteristic is controlled by 2 discrete inherited units. We now call those genes. He didn't know what they were called. And these are passed down from the parental generation to the offspring. Now, first and foremost, this really only works when we're talking about sexually reproducing species. So first off, understand Mendel is studying pea plants. Plants are sexually reproducing in most cases. In many cases, I shouldn't say all cases, but in many cases. Same thing with animals and other multicellular eukaryotic species. To be fair, this is not going to be the case for unicellular species that reproduce asexually. So, for example, asexually reproducing yeast or prokaryotic species like bacteria and archaea. They don't have these pairs of genes. So, in sexually reproducing species, the way inheritance happens is through the creation of haploid gametes via the process of meiosis. So he has no understanding of this at all at the time. We don't know what meiosis is. We don't know how gametes work. If you want to see what they thought like little human babies looked like and what sperm and egg looked like, look at the word homunculus. They literally thought that like sperm were just little tiny baby humans that made it into the mom, which is just kind of wild. So we know now that the reason why these gametes get shared the way they do is through the process of meiosis. So meiosis takes a diploid cell, and then through the process of reductive cell division, meiosis, we end up with haploid gametes, sperm and egg, okay? Then when two of these gametes fuse, one sperm, one egg, we end up with a diploid zygote, and half of the zygotic genetic information is made up of mom's information from the egg. The other half of the genetic information inside of that zygote is the paternal information that comes from the sperm cell. Then through repeated rounds of mitosis, you end up getting in humans, the trillions of cells that make up a human person, essentially. Same thing goes through there. And then when that individual reaches reproductive maturity, they go through meiosis and create gametes to make the next generation. But in reality, it's rarely that simple, okay? First and foremost, many characteristics, many of the things that we observe in people are controlled by multiple genes, all right? So for example, skin color, height, even eye color, which we used to hold up as sort of the model of discrete inheritance. These are not based on a single gene inheritance. They're based on polygenic inheritance, or the inheritance of multiple genes. One of the ways you can easily identify a trait that's not inherited discretely, but is inherited through polygenic inheritance, it's going to be something that is usually appears on a scale through continuous variation. Okay, so instead of being sort of one or the other, you can get a bell-shaped distribution of these particular phenotypic appearances. So just as an example, when we look at skin color in humans goes on a continuum from very light to very dark. And if you have two individuals who reproduce and make offspring, often their offspring aren't going to be at either end of their parent spectrum, but somewhere in the middle because of the way those pigment genes are inherited. When it comes to eye color, yeah, we can lump eye color into some broad categories: brown, blue, green, hazel. But even within those groups, you can see that there is variation. There are lots of different colors of blue eyes. There are lots of different colors of brown eyes. So while the main color is controlled by one gene, the variation within inside of that is continuous, which tells us that there are actually multiple genes that go into controlling eye color. So while it was really, and I don't want to discount what Mendel was teaching us, because understand, this is a guy that's just working in, Central Europe at a monastery growing pea plants, and he happens to examine discrete traits during that process. He's actually working against the blending theory of inheritance, which this seems to align with to a certain extent. But we now know that rather than being pairs of genes, in reality, many of the characteristics that we see are inherited through the activity of multiple genes. We call these polygenic traits. So we can see that Mendel's first law of inheritance is fairly readily broken in a lot of organisms. I use humans as an example there. Turning to his second law, the law of dominance, which basically states that you have multiple traits that can be inherited for a given characteristic, right? Multiple versions of that characteristic: eye color, height, hair color, whatever you want it to be. What Mendel would say is that you have a pair of genes that controls this. We're just going to discount the polygenic stuff for now. You have a pair of genes. If these differ, if they're two different versions of that gene, you are heterozygous for that particular gene. You have two different alleles, two different versions of that gene. What Mendel tells us is that one of those versions has to be dominant over the other. And that is the trait that is going to control the phenotype, the outward appearance. So I'll just remind us that what the genes say, we call that the genotype. So you can be heterozygous for a genotype. I have one dominant allele, one recessive allele. If we're talking about true dominance, which Mendel was, the dominant allele will always control the phenotype. So for example, if you have one gene for brown hair, one gene for blonde hair, you're going to probably end up with mostly brown hair because that's what your genes say. I'll go back to eye color, acknowledging that it's not strictly a single gene controlling this, but broadly speaking, if you inherit one allele for some version of brown eyes, one allele for some version of blue eyes, you're probably going to end up with brown eyes because the brown-eyed allele is dominant. It is entirely, completely dominant over that. But we now know that in many cases, it's not really that simple. We now know that there are several versions of this type of inheritance. First off, there's incomplete dominance. So incomplete dominance occurs when neither allele is able to dominate the other one. And in this case, when you end up with a heterozygote for incompletely dominant alleles, you end up in the heterozygotes with a unique intermediate phenotype. So for example, in this particular group of flowers, you've got the red allele and the white allele. Note they're written differently too. When we write completely dominant alleles, it would be like big R, little r, big W, little w. We don't do that here. We write C for color. That's how it's just the variable we've assigned. And then we write the allele information as a superscript. So CR, CW. This acknowledges that these are incompletely dominant with each other. And when you mix them together, when you get the heterozygote, you get pink, which is intermediate, right? Halfway between red and white, you get pink. This is something that the blending theory of inheritance, this old anachronistic understanding of genetics, would say like, yeah, that's what you would get. But where that falls apart is once you allow these F1s, these heterozygotes, the result of the P1 mating, if you allow them to either self-fertilize or mate with each other, you get the phenotypes right back out and you get the 1:2:1 ratio of homozygous red, homozygous white, and then the heterozygotes, which are all pink. So that's incomplete dominance. Co-dominance, on the other hand, is when you mix the two alleles together, you get them both in their entirety. So when we look at the ABO blood grouping in humans, we can actually observe a couple of types of dominance. So first off, we have to understand how this works. So when we talk about the ABO blood grouping, we're actually talking about a molecule on the surface of our red blood cells. And if you have the A allele, the A version of this, you produce a certain type of you produce the A the A molecule on the surface. If you have the B allele, you produce the B molecule on the surface of your red blood cells. And if you have the O allele, the O version of this gene, you produce neither A nor B. So your red blood cells are functionally naked with respect to these different antigens, okay? So what that means is if you are IA, or IA little I, you have type A blood. The one thing to notice, though, is if you have one A allele and one O, the little I allele, notice that it's big I, little I. That is conveying to you that actually this allele is completely dominant over this allele. So basically, whether you're AA or AO, you have type A blood because A is completely dominant over O. Same thing with B. If you're BB or BO, you end up with type B blood. But if you get one A allele and one B allele, you don't end up with A or B. You also don't end up with some sort of intermediate like A/B. Instead, you end up with AB. Your red blood cells will have both the A antigen and the B antigen on the surface of them, and you have AB blood. So what you need to notice with codominance, what separates it from incomplete is we don't end up with like a hybrid pink. We don't end up with a type C blood. We end up with AB, which would be the equivalent of ending up with flowers that are both red and white. So that's how this works. So with A and B, they are completely dominant over O, but A and B are co-dominant with respect to each other. So that's another type of dominance. So we've talked about complete dominance. That's what Mendel described. Incomplete dominance and co-dominance. The other thing we have to acknowledge that in some cases, the classic example here is rabbit fur, you end up with multiple alleles. So what you can see here is, note the big C's here. When you end up with big C's, you get brown. Then you've got these, notice that they're small c, so they're recessive to this guy. But when you end up mixing the CH or this the CH and the CH, you get these like intermediate phenotypes. So with multiple alleles, you have a lot going on. There's a lot of different ways, and it really depends on which alleles you're inheriting and the combination you get. So in other words, what we're saying here is dominance is not as clear cut as Mendel wanted it to be. It's a lot more complicated in reality. Okay. We also get to this also kind of goes down the list of dominance. This is something called epistasis. So epistasis is a very interesting part of genetics that has actually been utilized by geneticists to explore molecular pathways to help sort of order the way in which genes relate to each other. We'll give you an example of that in just a minute here. But first, let's talk about what epistasis is. So epistasis is the antagonistic relationship between two different alleles for two different genes. So remember, Mendel would say that these two genes, like this pair of genes that you have controls this particular characteristic, and that what happens at this is completely unrelated to what happens with other genes. And epistasis is going to show that that's not strictly true. So basically what we're saying is you can have a gene, you have an allele of a different gene that masks the phenotype of an entirely different gene. Okay? This might be tricky to understand, but we're going to give you an example here with mice. Okay? So with this particular example, there are two genes we have to talk about. We have gene A, which is a gene that's referred to as agouti. We'll explain what that means in a minute. And gene C, which encodes a separate protein that's called coat. Okay? Here's where it gets interesting. If we take two brown mice that are heterozygous, these are agouti colored mice, agouti. That's the color. That's why this gene is called agouti, because this gene agouti encodes the fur coat pigment protein. Notice that they're heterozygous at both the A allele, or for the A gene, and the C gene. So big A, big C, little c. When we combine them, we end up getting an interesting relationship. And here's why. If we're doing a standard dihybrid cross, you would expect to get a 9:3 to 3:1 phenotypic ratio, which means you would expect to get 9 out of 16 times both dominant phenotypes. Okay. In 3 out of 16 times, you'd expect to get one dominant, one recessive. In the next 3, you'd expect to get the first one you had dominant, that would be recessive, and the other one would be dominant. And then finally, you'd have the double recessive 1 out of 16 times. That accounts for your 16 potential variations. But we don't see that here. Instead, we get our 9 for agouti, which makes sense, dominant, brown. We get our 3 black, so recessive at the A gene, but some type of dominance out of the coat protein, that those mice turn black. But weirdly, these guys down here, we have we have homozygous dominant for A, heterozygous for A, and homozygous recessive for A. Yet no matter what happens, we get albino mice. So theoretically speaking, this is not what we should see. 3 of these guys have the dominant A allele, which should turn these mice agouti colored, right? Because that dominant A gives you that brown agouti coat coloring. But they don't. But the thing we have to look here is double A, big A, little a, double little a. But what is common to all 4 of these probabilistic outcomes, it's homozygous for the recessive coat protein, the coat allele. This is what epistasis looks like. Rather than giving the dominant brown phenotype as we should get, the fact that we are recessive, homozygous recessive for this coat gene makes them all turn white, which means this particular gene is controlling the output of this gene. Whatever's encoded by C is important for the expression of A. Once we've examined this more to understand this pathway, what we understand is that this particular gene encodes an enzyme that's required for making the protein that's encoded by gene A. What that basically means is that A can't function without C. By breaking this C gene, by breaking that protein, it doesn't matter what A says because we can't make it. The equivalent would essentially be saying we can make multiple cars, multiple versions of cars. And what happens at A happens here. But we need C to make the manufacturing process. Without having the assembly line working, it doesn't matter what instructions we get, there's nothing to assemble the car on. So this may say make an SUV, it may say make a van, it may say make a four-door sedan, but if we don't have the ability to produce the car in any way, shape, or form, it doesn't matter. So what this tells us is that if C develops a loss of function mutation, no protein will ever get made, and therefore the result is a mouse that's albino, lacking any coat protein whatsoever. In this context, what we would say is that gene C is epistatic to A, because whatever happens at gene C can dictate what happens to A, which is a really interesting genetic outcome. And what people began to realize is we can use this to understand how genes relate to one another. So here's an example. This is going to come out of an organism called Caenorhabditis elegans, which is a nematode. It's a very commonly used genetic model organism, and one of the reasons why we do this, why we use this organism, is it has a fully sequenced genome. It's very easy to do matings with, easy to handle, short generation time. There's a lot of great things about C. elegans. I use them in my graduate work and postgraduate work, which is why they're sort of like near and dear to my heart. But what epistasis analysis does is it can allow us to order genetic pathways. It allows us to understand the order in which signaling pathways work. It can help reveal relationships between what we thought were unrelated pathways. It can even help us identify which transcription factors are downstream of receptor activation. So let me give you an example of this. This is a signaling pathway. It's actually the insulin signaling pathway in C. elegans. We ordered this pathway first in C. elegans, and then we looked at human cells and said, oh yeah, it's basically the same, but a little more complex. So we gained our understanding of the insulin signaling pathway for mammals, humans, higher organisms, by studying it in worms, because we can do this kind of epistasis analysis very, very easily. One of the things that came out of this was, if you have, so this blue protein you're seeing right here, it's encoded by a gene called DAF2. Don't worry about what it means. It's C. elegans nomenclature. But this is the insulin-like receptor for worms. And what's very interesting is you can mutate this. You can't delete it. They need it. But you can damage it so it doesn't work very well. And when you put a loss of function or a reduction of function mutation into DAF2, worms live like 3 times longer. Like, it's pretty wild. The equivalent would basically be mutating it in such a way that humans would live for over 200 years. And it's even cooler than that. Not going into details, but it's not like you get to be 80 and then feel 80 for like 150 years. No. When you're 80, you feel like you're 30. Like, it extends your health span. And it's pretty wild. And there's a reason why so many people are investigating this in worms, right? But what was really interesting is, this signaling pathway, when this receptor is bound, it signals down through a big chain of other signaling pathways. This is called signal transduction. And it eventually converges on a transcription factor called DAF16. Basically, when DAF2 is broken, no signals get through, DAF16 is always active. So basically, what this means is when DAF2 is broken, DAF16, this transcription factor, binds to the DNA and basically is responsible for transcribing a bunch of genes that help worms live like 3 times longer. How do we find that out? Simple. We took a DAF2 mutant that lived 3 times longer. They did a big screen. They knocked out a bunch of other genes in C. elegans and found out that when you eliminate DAF16, you go right back to normal lifespan, which through genetic epistasis analysis tells us that DAF16 is epistatic to DAF2, which gave them the first clue that this particular gene, the protein that results from this gene, is somehow involved in this signaling pathway. They went through and then identified several other genes, AKT1, AKT2, PDK, AGE1, DAF28, found all of these other genes are involved. This is this particular analysis, this epistasis analysis is the basis for things that we call suppressor screens. So for example, DAF16 mutations suppress the result of DAF2, or enhancer screens, things that make things worse or exacerbate. So for example, if you take DAF2, they live 3 times longer. Is there another gene you can not make them knock out that make them live 4 times longer? That would argue in an enhancer screen, that would argue that particular gene is in a separate genetic pathway because it's additive. You add them together. So these are cool techniques I'll talk about in other videos, but keep in mind, epistasis is important. It helps us explore genetic pathways by describing how multiple genes can interact with each other in terms of the phenotype. The third law, Mendel's third law, is the law of segregation. It basically holds that offspring have an equal chance of inheriting one of 2 possible alleles from each of their parents. This is essentially why Punnett squares work, right? When we do those big Punnett squares, you put big, big B, little b from dad, big B, little b from mom, and then you basically just do the math, right? You move things around, and that tells you have a 1 in 4 chance of this, a 2 in 4 chance of this, a 1 in 4 chance of that. What we know in reality is this comes down to meiosis, tetrads, and sister chromatids. It's that process of meiosis where you start with during metaphase, you get these different variations, right? When you do that, when we do the independent or the random assortment during meiosis, right? They line up on the metaphase plate. You've got random assortment during meiosis 1, and you've got, the paternal chromosomes here, the maternal chromosomes here, and they go this way. And then for the next gene, maybe it's maternal, they go that way. So what that means is for each gene, there's a 50-50 chance that during meiosis 1 that the paternal chromosomes are going one way or the maternals are going the other. And basically, we can skip over all this because it holds up. From what we can tell, Mendel's third law of segregation actually holds up in most cases. There's no exceptions to this. In general, you have an equal chance of getting either your mom's chromosome 1 or your dad's chromosome 1. That's just the way it's going to happen because that's the way meiosis works. I do talk in my mitosis and meiosis videos about the fact that this random assortment is one of the ways in which genetic variation is introduced into species, and there you have it. His last law was the law of independent assortment. So don't confuse segregation and independent assortment. It's easy to do, especially because the law of segregation is supported by random assortment during meiosis. Don't confuse them. Independent assortment basically says that all genes are inherited independent of one another. All right. What you inherit on this gene is separate from what the inheritance of this gene. And in many cases, this works out. Okay, I just talked about a dihybrid cross. And I talked about how, you've got to account for all of the paternal combinations, all of the maternal combinations, and then you move them across. And we talked about actually in the epistasis analysis that we get, a 9, 1, 2, 3, 4, 5, 6, 7, 8, 9, double dominant. We get 1, 2, 3, 1 dominant, 1 recessive, 1, 2, 3. the other dominant, the other recessive, and then the double recessive, right? This is because the gene for P shape and P texture are inherited completely separate of each other. Okay, so the math works out because they're not dependent on each other. But in reality, some genes are actually linked. The way you know this is you do a dihybrid cross and you don't get your 9:3 to 3:1 ratio. Your 9:3 to 3:1 phenotypic ratio. What ends up happening with linkage comes down to chromosome structure. All right. So let's go back to meiosis. Let's remember the other part of meiosis that leads to genetic variation. It's the crossing over. Crossing over happens during prophase 1 of meiosis. And what happens is the chromosome, one of the paternal chromosomes in your sister tetrads, will cross over with one of the maternal chromosomes in the other pair of sister chromatids, and they will exchange information. And you end up with a recombinant chromosome in each of the sister chromatids. This right here is why most genes are inherited independent of each other. But genes that are extraordinarily close to each other on a chromosome often cross over together. And as a result, they essentially act as a single megagene, which means if mom's allele for C is very close to mom's allele for B, when C crosses over, B crosses over too. In other words, it's very, very hard to get those alleles to separate, and as a result, you often get two entirely unrelated genes being inherited together. We call these linked genes. This was actually understood by an American geneticist whose last name was Morgan, and he began to use this as a technique for exploring chromosome structure. And basically, you look at your results from your dihybrid crosses and see what your phenotypic ratio is. Unlinked genes will always have a recombination frequency of 50%, which he dubbed 50 centimorgans. Genes that are closer together will have a recombination frequency that is much lower than that. And as a result, instead of getting a 9:3 to 3:1 phenotypic ratio, you start to get ratios that are much closer to the 3:1 ratio you would get from a monohybrid cross because they're acting like a supergene. So if you think two genes might be linked, the way to study this is do a dihybrid cross. If you get 9:3 to 3:1, you are looking at genes that are unlinked. If you get a 3:1 phenotypic ratio, they are completely linked. And if you get some number in between, you now understand that you have genes that are partially linked. They're not right next to each other on a chromosome, but they're close enough where in some crossing over events, they go together. And we can use that as a way of understanding chromosome structure. All right. So for example, if you end up with a 3:1 phenotypic ratio, chances are for genes B and C, chances are B and C are right next to each other on the chromosome. But if you get a 9:3 to 3:1, they're very far apart. If you get a recombination frequency less than 50%, but less than 50%, that's an indication of how close they are on the chromosome. The smaller that recombination frequency, the closer they are. So if one pair of genes has a 10% recombination frequency, the other has a 5% recombination frequency. The ones that have 5% recombination are closer together than the ones with 10%. So how do we use Punnett squares to predict inheritance? Well, again, Punnett squares are good at predicting this because of math. Genetics basically comes down to probability, and Punnett squares are just a visual method of calculating expected patterns of inheritance. This is easy when we're dealing with one gene or two genes, maybe even three, but let's put it this way. A monohybrid cross is going to have 4 squares. Oops, I went the wrong way. Our dihybrid cross has 16 squares, which means our trihybrid cross is now up to 64. And if we go with 4 genes, we're up to 128. And at some point, this just becomes untenable. Okay? Now, again, if we're dealing with an instance of linkage, if we're dealing with an instance of epistasis, we're going to have to reevaluate how we do this because, down here, you're going to have your albino mice because of the epistatic effect. Or you're going to have this 3 to 1 phenotypic ratio because you're dealing with linkage. Okay, so linked genes will deviate. Epistasis can lead to unexpected results. But let's just talk about normal genes. What can we do? Instead of having to account for all this, we can simply use math. Okay, so when we're in the world of genetics, most geneticists are not going to sit down and do a Punnett square unless it's literally to demonstrate to a graduate student or a new worker in the lab how this all works. We really want to know probability. Okay, and that leads us to 2 rules. First is the sum rule. This determines the probability of either of 2 mutually exclusive events occurring. This is our or rule. This will happen or that will happen. What is the probability of this happening or that? Mutually exclusive means if one happens, the other cannot. Okay? So I can either have purple flowers or white flowers. Can't have lavender, can't have both. The product rule determines the possibility of both mutually exclusive events occurring. Okay? Both have to happen. Okay? So this is our and rule. And understandably, when we multiply fractions, we get smaller numbers than when we add them, right? It's much more challenging for 2 things to happen than one thing to happen, or than one or the other to happen. So let's examine a way that this can work. So for example, what is the probability that the progeny will inherit a dominant B allele from at least one of their parents? Let's say we're looking at a heterozygous situation here, all right? What is the probability that you will get one big B from either of the parents? Well, there's a 1 in 4 chance that they get the big B from mom. There's a 1 in 4 chance that they get the big B from dad, right? 4 potential outcomes. Again, we are looking for only one. Our sum rule would say that there's a 1 in 4 chance that you get just mom's big B. There's a 1 in 4 chance that you get just dad's big B. And as a result, there's a 2 in 4 chance that you're going to end up heterozygous with a dominant allele. Right? 2 in 4 is 1/2. The product rule then will be, what if you inherit it from both parents? Okay? What are the probability you get it from both? In other words, what's the probability of being homozygous at the dominant allele as opposed to being heterozygous for the dominant allele? Here we go, well, there's a 1 in 2 chance that you get it from dad, a 1 in 2 chance that you get it from mom. Multiply and we get 1/4. And if you look at the Punnett square, yep, there it is. There is our homozygous for the B allele. Here's our heterozygous, sum rule, homozygous. Okay. Again, what if you get the big B from mom or from dad? 2 out of 4. What if you get it from mom and dad? 1 in 4, right? Or and. All right. So that's what that's going to look like. All right. I don't know why it's duplicated over here, so don't worry about it. So again, I will reiterate that genetics is all math and probability. If you're looking for a way to practice this, here is a dihybrid cross for a Punnett square. Can you do the math to figure out all of the different genotypes in here? Can you use product rules to figure out what are the chances of you getting the big R, little r, and big Y, big R from both parents and the big Y from both parents, right? What are the odds of this? Well, for big R, it's 1/2 times 1/2 is 1/4. For the mom, it's big Y. From the dad, it's big Y, little y, another 1/4. Multiply them together, 1 out of 16. There's a 1 in 16 chance that you're going to end up being homozygous for the dominant allele for both of these particular genes. Notice it's the same probability of inheriting both little r's and little y's from mom and dad. Okay, 1 in 16. All right. So that's a good exercise if you're looking to practice these types of things and understand this. We've got to get more comfortable with the math as we get higher up in genetics because it's just not feasible to do all these Punnett squares. So I hope you enjoyed this introduction to Mendel's laws, how they're kind of broken, how we can use these things to actually better understand molecular pathways and genetic pathways in living organisms. Thanks for tuning in, and I'll talk to you all real soon.